A warm-hearted greetings to all watching this presentation. I'm here to present research work on formulation and evaluation of modified Pulsincap drug delivery system of Ketoprofen for chronotherapy of arthritis. First and foremost, I would like to thank DRPI for giving me this wonderful opportunity. Starting with the introduction. Pulsatile drug delivery is a system in which rapid and transient release of the active molecule occurs in a short period of time immediately after the lag time. It implements the concept of chronopharmaceutics in which the drug release is designed according to biological rhythm of the disease. A Pulsincap system, it consists of water-insoluble capsule body filled with the drug formulation and plugged with a swellable hydrogel at the open end. Upon contact with the dissolution media or GI fluid, the plug swells and comes out of the capsule after the lag time followed by the drug release. Coming to the need of the study. Arthritis is inflammation of one or more joints causing pain and stiffness that worsens with the age. Morning stiffness and pain are the main problem. It's a chronobiological issue. Though ketoprofen is an effective NSAID, also it has some drawbacks. That is, it has short biological half-life of 2 to 3 hours, because of which it needs to be administered in 3 to 4 divided doses. Further, also it causes upper GI tract bleeding on recurrent administration. Hence, the objective of this study was to design and characterize an oral site-specific pulsatile drug delivery system containing the ketoprofen, which can be targeted to colon in the pH and time-dependent manner to modulate the drug level in synchrony with circadian rhythm of osteoarthritis and rheumatoid arthritis. Coming to the formulation design of ketoprofen micro capsules. So, the material used in the preparation of the Pulsincap system is shown on the left-hand side. Now, coming to the formulation code, 6 batches from F1 to F6 were prepared with different proportions of core to core material as shown in the tabular column. F1 and F2 contain the drug with polymer Utrazid RL and RS100. F3 and F4 contain the drug with Utrazid RL100. F5 and F6 contain the drug with Utrazid RS100. Let's see the method and the result. Initially, the first step was preparation of the micro capsules containing ketoprofen. The method used was emulsification solvent evaporation method. Here, Utrazid L100 and RL100 and RS100 in the ratio 1:2 were dissolved in 10 ml of acetone to form a homogeneous polymer solution. Then 1000 mg of ketoprofen was added to the polymer solution and mixed thoroughly. The organic phase was slowly poured at 15 degrees centigrade in the 150 ml liquid paraffin containing 1% weight by volume of span 80 with stirring at 1200 rpm to form the smooth emulsion. The micro capsules were subjected to decantation for collection and it was washed with petroleum. Either further, it was dried at 40 to 60 degrees centigrade for 3 hours. Hence, micro capsules were produced. The next step is the micro capsules equivalent to 100 mg of ketoprofen were accurately weighed and filled into the formaldehyde treated bodies of hard gelatin capsule. The body was plugged with HPMC hydrogel at different concentration. The capsule body and the cap was sealed with 5% ethyl cellulose ethanolic solution. The sealed capsules were completely coated with 5% cellulose acetate phthalate to prevent variable gastric emptying. Hence, the whole system produced a modified Pulsincap system. Next, the evaluation of microcapsules and Pulsincaps were carried out. The first and foremost study was FTIR spectroscopy to understand the compatibility between the drug and the polymer. From the spectra of ketoprofen, physical mixture of ketoprofen and polymer and ketoprofen microcapsules, it was observed that all characteristic peak of ketoprofen were present in the combination spectrum, thus indicating compatibility of ketoprofen and the polymer. It was followed by scanning electron microscopy. This was performed to characterize the surface of the formed microcapsule. Formulation 1, 3 and 5 showed the rough surface, but they were spherical in shape, whereas formulation 2, 4 and 6 produced the microcapsules with a spherical, smooth surface and discrete. Next, the mean particle size or average particle size of the microcapsules were determined by optical microscopy technique. So, the average particle size of the microcapsules ranged from 628 micrometer to 870 micrometer. The mean particle size of the microcapsules increased with the increase in the polymer concentration. This was followed by frequency distribution analysis. All the formulations showed normal distribution bell-shaped curve. It was observed that as ketoprofen to polymer ratio increased, the mean particle size of ketoprofen microcapsules also increased. This was followed by flow property. The flow property was determined from angle of repose value. As tabulated, it was observed that all the batches were found to be fit in respect of flowability as theta value ranged between 22 to 33 degrees. The next was percentage drug entrapment efficiency. So it was observed that the entrapment efficiency increased with the increase in the polymer concentrations. Hence, it can be inferred that there is proper distribution of ketoprofen in the microcapsules, and the F2 formulation showed the entrapment efficiency of 94.5%. This was followed by in vitro dissolution profile study. So, from the in vitro release study carried out for 12 hours, it was observed that drug release from the formulation decreased with the increase in the amount of polymer added in each formulation. So, to understand the mechanism of drug release, various kinetic models were studied like zero-order release kinetic, first-order release kinetic, Higuchi-matrix release kinetic, and Peppas release kinetic. Based on the kinetic release study and from the R-square value, it was found that all the formulation followed zero-order kinetic. From the Higuchi equation, it was found that the drug release followed the diffusion mechanism. To know the type of the diffusion mechanism, Peppas equation was studied. From the N value, it was concluded that the drug release happened through non-Fickian diffusion. Then the modified ketoprofen Pulsincap were evaluated for weight variation studies. All the the weight of all the capsules were within the specified limit. Further, the thickness of the cellulose acetate phthalate coating were also determined, and the values are tabulated in the tabular column. This was followed by in vitro release profile of the pulsatile drug delivery system containing ketoprofen, which was carried out for 24 hours. So, from the in vitro release studies of pulsatile drug delivery system, it was observed that there was absolutely no drug release in the simulated gastric fluid for 2 hours, that is in the pH 1.2 medium. Then the negligible amount of the drug release was observed in simulated intestinal fluid of pH 7.4, where dissolution was carried out for 3 hours. In the remaining hours, dissolution was continued in simulated colonic medium, that's of pH 6.8 phosphate buffer, and the release was found in 24 hours in controlled manner depending upon the quantity of the polymer plug. Then it was followed with the DAC study, differential scanning calorimetry. The DAC of the drug alone showed sharp peak indicating the drug is in crystalline state. DAC of the ketoprofen microcapsule showed the disordered crystalline state indicating good compatibility. It was followed by stability studies. So from the stability studies, it was seen that all the formulations were quite stable because there was no significant change in the physical properties and drug content of the capsule. From all the result and data obtained, coming to the conclusion, the design formulation produced consistent pulsatile drug release for a period of 2 to 24 hours as per the requirement of chronotherapeutical drug delivery system. Further, the Pulsincap system of ketoprofen was found to be promising for colon-specific drug delivery and chronotherapeutic management of arthritis. Hence, the design formulation has a very good market potential. The future scope involves carrying out in vivo studies, which is already in process. Thank you.